Yes, sir. Start. A very good morning to one and all. Welcome back to GIAN course. This is the day 4th of the GIAN course series, and today we'll start by Dr. Hitesh Handa for this lecture 1 of the day 4. So please.
यहीं रख देते हैं. We can keep it here. Oh, pocket. Okay.
अगर मोमेंट में भी रहेंगे.
Okay. Okay. ठीक. Thank you. Great. Thank you. Okay. Good morning, everyone. Let's start with our class. So, if you remember, yesterday we talked about protein adsorption on the surfaces, right? How fibrinogen adsorbs, and now fibrinogen has receptors on which platelets can adsorb and get activated, and it can also lead to infection, which will be talked tomorrow. But today we will talk about blood clotting, that how after proteins adsorb, how platelets can stick to the surface and get activated, which can lead to clot formation. Okay. Okay, so blood is a living tissue that many medical devices come in contact with, right? If you have an implant that's going in bloodstream, for example, catheters, vascular grafts, stents, extracorporeal devices where blood is going outside, touching the tubing or foreign surface. Now blood is coming in contact with foreign surface. Blood is a mixture of plasma, solution of water, salts, proteins, different types of cells, platelets, all mixed together in blood. Okay. All blood cells originate in the bone marrow via a complex process termed hematopoiesis or blood forming. So bone marrow, the blood is formed inside the bone marrow. At birth, marrow throughout the body is red and produces red blood cells, but slowly it starts turning yellow. And in adulthood, this blood cell production tends to be found in femurs, pelvis, sternum. That's where your blood cells are getting formed. Now, so if I draw your blood right now in a syringe or a vacutainer, what do you think what will happen if I just draw if I have an empty syringe and I draw blood? What do you think what will happen to the blood? But blood will be in the syringe, but will it be liquid or will it get solidified? Liquid, right? But why liquid? If my syringe has nothing in it, so you know when you go to a doctor, they draw your blood, the blood is liquid because there is something in that tube or syringe that's preventing blood from clotting. Okay, and that's called anticoagulant. If you don't have anticoagulant in that syringe or vacutainer, blood will clot immediately. Okay, so if I have an empty syringe and I draw your blood right now, within seconds the blood will clot. It won't be liquid, it will be solid. And the only reason it's not clotting is because those syringes have anticoagulants in them, those vacutainers have anticoagulants in them that prevents blood from clotting. So now why do you think blood clots? You know, when it's in the body, it's just fine, it's flowing properly, but as soon as I draw it in a syringe or vacutainer, why is it clotting? B.Techs, you all are B.Tech, right? Undergrads? Why do you think blood clots? It's not very desirable for us.
In case of any injury or wound, if the blood tends to flow out of the body, it needs to
Yes.
be stopped.
Great. Yes. So blood clots because if you get an injury, you want the blood to clot. If blood won't clot, we will bleed to death, right? So now let's say I had the anticoagulant in the syringe, I draw the blood. Now if you centrifuge blood, okay, what will happen is blood will get separated in these different components, okay. Almost half of it, or approximately half, let's say, it will be red blood cells, white blood cells, and then the top, which will look more yellowish in color, will be plasma that has nutrients, water, even some platelets, and then the bottom part, heavier cells, are red blood cells, white blood cells. Okay. So this top part is plasma, bottom part is all the other cellular components, red, white blood cells. Now, we will talk about it later, but I have a little interesting thing to tell you. So what we do is when we centrifuge blood, we get platelet rich plasma, okay. This plasma has lot of platelets in it. Now, let's say I take with a pipetter, I take all these platelets in a separate tube, platelet rich plasma, this liquid I take in a separate tube, and now I spin it very hard at a very high speed, so all the platelets settle down and form a pellet, and now you are left with platelet poor plasma, right? First it was platelet rich plasma, we centrifuged it much harder, now we have platelet poor plasma. Approximately in our blood, we have approximately 3 × 10⁸ platelets per ml. Okay. Now that's platelet rich plasma. It can be 4 × 10⁸ also, but approximately that number. When I centrifuge it so hard, when I get platelet poor plasma, what do you think, what's the platelet count in platelet poor plasma?
10⁵, 6, maybe something of that order.
Anybody else? So when I started working in the field for first few weeks, I used to think platelet poor plasma is 0 platelets. Okay. But then I found out that actually platelet poor plasma still has approximately 10⁶, 10⁶ platelets. So it's only 10 times less than platelet rich plasma. Okay. And later on, I think maybe tomorrow or next week, we will talk one of the reasons why we in our experiments, we need platelet rich plasma also and platelet poor plasma also. We will talk about it later, but that was just an interesting thing. Okay, now blood has lot of different proteins. Some of the proteins are albumin, IgG, fibrinogen, prothrombin. Yesterday we talked about how fibrinogen is the main culprit on which platelets like to get activated and stick to. And fibrinogen is almost 10 times less than albumin. So this is just approximately showing concentrations of different proteins. Now, blood has red blood cells, white blood cells, platelets. For clotting purposes, platelets are very important. Okay, but let's talk a little bit about red blood cells also. So red blood cells are also called erythrocytes. Erythro means red appearance and cytes are cells, so red cells. Erythrocytes have no nucleus, so they cannot proliferate. Red blood cells play minimum role in wound healing and in blood material interactions. because that activation clotting that's happening, red blood cells are not playing that much role in it. That's platelets' job. They will be used to demonstrate basic biological engineering concepts. Red cells contain hemoglobin, and that's for oxygen transfer and carbon dioxide removal from the body. That's red blood cells or erythrocytes. These erythrocytes can survive for approximately 120 days, and then they are removed by spleen and bone marrow and get cleaned from the body. Oxygen demands of body can upregulate production of erythrocytes. So if you are on elevated places where your body is little bit lacking in oxygen, signal can be given to the body that, hey, I need more oxygen. They can make more red blood cells, so more oxygen can be carried. And this is like at low tissue oxygenation stimulates the production of red blood cells by increasing level of erythropoietin, which actually helps in within minutes to hours, it can upregulate these red blood cells so that that oxygen demand can be taken care of. So this shape of these red blood cells is bioconcave, and they are 7 micron in size. And this shape is very special because it has its maximum surface area to volume ratio, and red blood cells are extremely deformable, which they should be. The reason is when blood is flowing through tiny blood vessels, sometimes the size of the blood vessel is approximately 7 micron, and you have 7 micron red blood cell size. So if they are not deformable, they can clog the blood vessels, which we don't want, of course, right? This is just from internet, some pictures of red blood cells. Now, like I said, capillaries have diameter of 5 to 7 micron. So this circulation, we want them to deform easily so they can keep flowing in these tiny blood vessels. Another concept is hematocrit. So whole blood, plasma and cellular components is a non-Newtonian fluid, right? So because viscosity varies with applied shear rate, it's non-Newtonian. Hematocrit is percentage of volume occupied by red blood cells within the blood. And for men, it's approximately 46%. For women, it's approximately 38%. So when you are centrifuging, that volume of that red blood cells is approximately 46%, and that's the hematocrit. And for women, it's around 38%.
So does this depends upon hemoglobin?
Yes, so red blood cells, right? Yeah. So non-Newtonian characteristics of blood flow are due to flow behavior of red blood cells. So you guys know Newtonian and non-Newtonian fluids, right? And some example of non-Newtonian, what else do you know is non-Newtonian? Cornstarch. Yes, right? Because, so water is Newtonian or non-Newtonian? Right? Because you apply more pressure, let's say you have like that water gun, you apply more pressure, more water is coming out. But for cornstarch, if you are stirring it slow versus fast, its viscosity is changing. It's not like a constant viscosity, right? Okay, so another concept in blood flow is this rouleau. So at low flows, these red blood cells can stack one over another, and I'll show you, I can quickly show you first. So these are like rouleau formation. So with interactions with each other, weak protein membrane interactions, these red blood cells can stack over each other, and this is known as rouleau. As shear rate approaches zero, roulae aggregate into masses and start behaving like a solid. But as the shear rate increases, protein membrane interactions break and viscosity of the blood decreases and the flow becomes again fine. So there is this concept of roulae also. Now, okay, let me ask you one question. So, do you guys know what is a hypertonic solution or hypotonic solution or isotonic solution? Okay, can one of you talk about it? What is a hypertonic solution?
So if the solute is suspended more in the environment than the internal component of the cell, if that is the case.
Yes.
Concentration is high in outside environment.
Yes. So, always for these biological studies, you can imagine if you are working with this blood or red blood cells and your or platelets, if your solution is hypertonic or hypotonic, meaning you have more salt concentration outside the cells or less concentration outside the cells, either water will trend to come out of the cells to dilute it or try to go inside the cells to maintain equilibrium. Now what will happen is you are either shrinking your cells or you are swelling them so they can actually burst and lyse, and both are undesirable for us, right? Both are not what we want. So we want to work with isotonic solutions so that the concentration is approximately same, so there is no swelling or lysis of the cell happening. Okay, now it's more interesting. So thrombocytes or platelets are also called thrombocytes. Why? What is thrombocytes?
Fragmented part.
Yes, so first, platelets are made up of mega creatoblasts which break down into platelets, but they are also called thrombocytes because they can lead to thrombosis, and thrombosis is basically clotting. Okay, so for wound healing, for blood material interactions, red blood cells is not that important, but platelets are very important because they are leading to clot formation. Okay, size of these platelets are 2 to 5 micron. Platelets have no nucleus, so they cannot proliferate just like red blood cells. Lifespan of platelets is 8 to 10 days. What's the span of red blood cells? 120 days, right? So this is like almost 10 times less, more than 10 times less. And then inactive platelets that are getting old, they are cleaned out by spleen, by macrophage that are scavenging it and just taking care of it and cleaning the blood. Now, platelets, when they are in our blood, they are very quiet and they look like smooth. Once they start getting activated, they start looking like that. Okay, and what is activation of platelets? It's like irritation of platelets, right? So when you have a foreign material, either blood is coming outside in a tubing or you're putting a catheter or medical device, blood is seeing that foreign surface, it starts sticking to the surface and then it starts getting activated like that. Once it starts activating, it's grabbing with all these little legs, it's grabbing to the surface and it starts releasing certain chemicals to tell other platelets that they need to come and help them aggregate and clot even more. And that's like very smart of the body, right? Because if there is a cut and you're badly bleeding, some platelets are coming and trying to stop it, but they are not enough. So they are sending signal to other platelets that come, there is your need, and they come also and start to stick in that area also. And then when they are activating, lot of things are going to happen. I'll tell you in the next few slides. but this is just an image showing quiet versus activated platelet. Now, for platelet surface interactions or blood surface interaction, a lot of things are happening, right? A reaction without adhesion. Platelets are platelets are flowing in the blood, they see a foreign surface, they adhere to it, then they spread it, then they start releasing these chemicals, they start aggregating more, and then they can embolize also. What was embolism? We talked about it yesterday.
So it was clotting or
Detachment. So you have something, yes, it's clotted, but then it gets detached to go down the bloodstream somewhere.
what gets detached?
It can be, so embolism is this phenomena. It can be clot, it can be a biofilm like bacteria that has
Mesh
aggregate.
that fibrinogen forms.
Yeah, whatever is attached, now it's detaching and going in the bloodstream.
Detached from the clot side, right?
Yes.
And so what activates the platelets?
That's the next 1 hour of talk, right? So, but the short answer is, see, yesterday we talked how proteins come and stick to the surface, right? And when proteins are sticking to the surface, they are spreading, and the receptors on the protein platelets are detecting and they are attaching to it. That's a short answer, right? But when you have a wound or a cut, then it's a little bit different mechanism. Okay, so like we said earlier, one of the main roles of platelets is to plug the holes, right? When you have a cut in the body. Platelets do not adhere to normal endothelial lining. So endothelial cells are lining the blood vessel. Platelets are fine. They are just flowing through that without sticking to the surface. But when there is a cut, now something is there that's telling the platelets that you guys need to start activating and start clotting, right? Okay, so platelets are small light blood cells. Platelets roll along endothelium in a normal vasculature when everything is fine, not activated by normal endothelial cells. Formation of platelet plug, when platelet plug happens, platelet adhere, platelets get activated, and now a little bit we are starting to talk. There is this expression of receptors called GP2B3A. These receptors that were hidden in the platelets now start coming out. Okay. Can you guess approximately how many GP2B3A receptors are present on each platelet? How many? Yes. What were you saying? So approximately 60 to 80,000. So imagine one platelet that is 2 to 5 micron in size can have 40, 50, 60,000 GP2B3A receptors on it. So you can imagine the nature has designed platelets so well that there is no way to miss the receptor, because these receptors bind to fibrinogen, the protein that's acting like a glue between other platelet. So you know, if there is a cut, and we'll go slowly through it, if there is a cut, there is a platelet, it's activated, GP2B3A receptors are out now on the platelets, they are binding to fibrinogen, because fibrinogen is also floating in the blood, and now the other end of fibrinogen is binding to another platelet. Now that platelet has more receptors, they are binding to fibrinogen, more platelet, so platelets are binding through fibrinogen glue in the middle just to stop bleeding, right? So now little bit another player that comes is collagen. So when you get this cut, collagen gets exposed, and this collagen starts telling platelets and blood components that this is not normal. Before that, there was like healthy endothelial cells, blood was flowing just fine. Now there is a cut, collagen is exposed, it's telling blood components there is something wrong, we need to stop this. So let's read, because adhesion is important to platelet function, platelet membranes contain glycoproteins, GP2B3A receptor. GP2B3A receptor can interact with this RGD sequence that's present in collagen and fibrinogen, right? And then there is another receptor also which we'll talk, GP1B I think, but that's also present on platelets and that interacts with the collagen also. But this RGD sequence we talked yesterday is present on fibronectin and fibrinogen. And that's the RGD sequence getting recognized by GP2B3A also. Now collagen is present in the base membrane in the vessels, which is usually covered with endothelial cells, endothelium layer, but once the cut happens, now it's exposed and now platelets are recognizing that. If endothelium is damaged, collagen is exposed, platelets adhere, other proteins in blood have high affinity for collagen. Once platelets adhesion occurs on the surface, the shape of platelets change and coagulation cascade is activated and blood clots start forming. Okay, we will talk about coagulation cascade in next few slides, but platelets shape change is the activation of platelets. Okay. Now coagulation clotting, hemostasis of site of injury. So if there is an injury, body wants that injury to be healed, take care of the injury so that blood can start flowing properly and everything is normal again. Now some of the main players when injury happens is clotting factors, platelets and vessel wall. Okay. Now I said coagulation cascade, right? Like blood clotting process. There are two different types of coagulation pathways. One is called extrinsic pathway, one is called intrinsic pathway. So you can think one is extrinsic pathway, one is intrinsic pathway, but then those pathways become same and it's a common pathway. Okay. How I remember it is, extrinsic pathway is like an injury. So external, you have a cut, it's extrinsic pathway. And when you get a cut, platelets have big contribution, right? Because if platelets need to recognize that cut and start plugging immediately, otherwise you will keep bleeding. Intrinsic pathway, I remember, is intrinsic, internal. So you are putting an artificial device in the body. And now fibrinogen plays a bigger role, because when fibrinogen sees this foreign surface, it sticks to the surface, and that's what platelets recognize and start getting activated. Okay. Now, like little bit process. Collagen is present in subendothelial. When everything is fine, it's hidden. But when you get a cut, it gets exposed. Platelets adhere to collagen mediated by von Willebrand factor or VWF, released from endothelium and platelets. So platelets are also a little bit releasing it, endothelium layer is also releasing it. Platelets start adhering to collagen via VWF, which is acting as a glue between platelets and collagen. So collagen gets exposed, Platelets want to adhere to that collagen. VWF is acting as a glue between them. Activated platelets, I earlier said, release some chemicals. So they release ADP, VWF, thromboxane, and all these are activating more platelets. All the platelets that were passing by, they see these chemicals released, and that's a signal for them to go there and help the other platelets. Platelet-platelet interactions start with GP2B3A receptors, fibrinogen, and VWF act as glue. So VWF is acting as a glue between collagen and platelets, fibrinogen is acting glue between platelets and platelets, and they all are trying to get this clotting going. And the same thing in little bit. So vascular injury, endothelial cells, collagen and tissue factor get exposed, platelets get come to the spot, they start getting activated. When they are activated, they start releasing ADP and thromboxane. When they release thromboxane and ADP, it really results in a clot formation. Okay. Okay, who can tell me the process now? If there is an injury, what happens?
So the collagen in the tissue
No, not you, somebody else. Okay, who will tell? It's easy, right? See, nobody will look at me now, but I used to do the same thing. Okay.
So when we get a cut, the plasma, the platelets combine and start the clotting of the blood to stop the bleeding.
Okay.
So what we have to explain, actually I forgot the main point.
Okay. So if like the whole process, when there is a cut, what's happening?
Platelets come, they release ADP, adenosine diphosphate, and thymosin. They attract more platelets, they activate, they have some legs moving.
So how do platelets know that they need to get activated?
They have receptors.
Fibrinogen mesh kind of a thing, and platelets got trapped into that mesh.
Okay, no, first, if platelets are just flowing in the blood,
Yes.
how do they know that they need to get activated or clotting should happen?
There are some cofactors like calcium ions and some other factors as well. So that is how they got the message that they have to block the passage or clot the blood.
Okay, so you're saying there is calcium ions. But how are calcium ions coming?
They are the cofactors of other clotting factors like, I don't remember the name right now, but they are basically, these ions are the cofactors for the blood clotting factors actually.
You are telling me everything that I'm going to teach, but whatever I taught, can somebody tell me in simple language?
So the collagen,
Gets released when there is a cut on the membrane of the cell. As the collagen is released, the receptors on the platelets, it gets attached to that collagen. I guess there is one receptor. So when they attach, then the platelets sheet changes and it gets activated. And then various factors like the VWF that acts as a glue between collagen and platelet will join them and just help them to stop the blood flow when there is a cut. And
Okay, some things were missing, but some now you can. Yeah.
usually the platelet is free flowing in the blood and it does not has any binding affinity with endothelial lining that we have. So when we have a cut, the endothelial lining is it's removed from there and the collagen and tissue factors are exposed. So due to that exposure, now the platelets in the blood has a binding affinity with its receptors to the collagen, and then the whole process kickstarts.
Yes. So once that collagen is exposed, platelets recognize it, and then VWS is getting released from that cut also. So platelets want to go there, that there is something wrong. They are like guards going in the blood, right? So something is wrong, and now when they see that there is a problem, they stick to collagen. When they stick to collagen with VWF, they get activated. Now they are sending signal to other guards that come help us, and that is thromboxane and ADP and VWF, and the other guards come and help. Fibrinogen in the blood is getting attracted to that area and getting bind to this GP2B3A receptor. More platelets are coming, and this resulting in clot formation to plug that cut in the body. Okay. It is confusing, really. I mean, it's not your fault. It is confusing, and remembering all this is little bit. But you wait 2 slides and you will see the real confusing thing. Okay. Okay. So basically, GP1B on platelets and collagen and VWF is interacting. Activated platelets and activated platelets, fibrinogen is the glue between them. Okay. Now we talked a little bit about that extrinsic pathway, right? External injury, that's what's happening. Now let's get a little bit more confusing. When coagulations happen, there are coagulation factors in the blood also, right? And here are some of the coagulation, here are the coagulation factors. So fibrinogen is so important, right? It has to be factor 1, okay? It's important. And it's okay if you don't remember these names of these factors, okay? Prothrombin is factor 2, tissue factor is factor 3, then factor 13 is very important because that's fibrin stabilizing factor. Before I go to the next slide, which is going to be even more confusing, let's talk a little bit what's happening, right, in the blood. So when there is a cut, we want fibrinogen to work and form clot and cross-link that clot. So what happens when platelets getting activated, fibrinogen is binding to them, it's still a loose clot. We need something cross-linking this whole thing, okay? And that cross-linking thing is called fibrin, okay? Or fibrin strands. Fibrin comes from fibrinogen. But when we are in the blood, fibrinogen is fine. It's protected, it's not causing any clotting. But when this clotting starts happening, fibrinogen gets converted to fibrin, and fibrin starts forming strands of fibrin, and these strands of fibrin start getting cross-linked to stabilize that clot. Okay. But for all that to happen, let's go to the next slide. This happens. Okay, so what happens is when there is a trauma or injury, factor 7, so these factors are proteins or enzymes activating. Factor 7 gets converted to activated factor 7, and that activated factor 7 will cause factor 10 to get activated. Activated factor 10 converts prothrombin was factor 2 to thrombin. And the role of the thrombin is to convert fibrinogen to fibrin. And now once fibrin is formed, it gets converted to cross-linked fibrin clot with the help of activated factor 13. So it's little bit confusing, but lot of things are actually happening. So when factor 10 is getting when prothrombin is making thrombin, thrombin is activating, if you see this arrow, thrombin is activating factor 5 also, which is helping to convert more prothrombin to thrombin. This is also activating factor 8, which is also helping convert more factor 10 to 10, activated 10, which is helping prothrombin to get converted to thrombin. I don't expect you to learn this for your exam, okay? Because if I don't look at it for few weeks, even I forget. So I cannot expect you to learn it. The point is, this is why it's called coagulation cascade, because it's like cascade of events happening, and you can see it just gets multiplied so fast. And it makes sense also, because if there is a bleeding happening, you want this to happen very fast so that we can just stop that bleeding from happening, right? But there is another interesting thing happens. If you see these little bit red arrows, red stuff here, body is so smart that it's starting the negative feedback loop also. They are already preparing how to stop this from happening also. Because see, if this clotting keeps going on, that's a problem, right? So thrombin is starting to make antithrombin also. It's helping in formation of this, which can release protein C, so that the negative feedback loop, plasminogen getting converted to plasmin, I'll talk about in next few slides. But body is actually preparing that as soon as this gets taken care of, let's start slowing this process also, so this clotting stops also. Okay. Another interesting thing here is, so antithrombin binds with thrombin, and once it binds with thrombin, now fibrinogen is not getting converted to fibrin. And guess what? So what are some common, and what is a common anticoagulant? Very common anticoagulant that we have heard about it. Huh? Heparin, right? Heparin is a common anticoagulant. How heparin works? Heparin binds to antithrombin, and when heparin binds to antithrombin, antithrombin becomes even more attracted to thrombin and binds more easily to thrombin. Okay, so that's why when people, we don't want them to clot, you can give a heparin injection, or they are going through cardiopulmonary bypass, or they are going through ECMO or dialysis. Heparin is given so that antithrombin thrombin interaction can happen, so blood won't clot. Okay, we will talk about this little later. That's why one way to prevent clotting on medical devices is also, there are devices that have immobilized heparin on the surface, so that when blood is flowing, that immobilized heparin on the surface is preventing blood from clotting. Okay. Oh, sorry. So this is one pathway, that's extrinsic pathway, this is intrinsic pathway, and they both lead to common pathway. Okay, but just remember, it's not very that, okay, you are just coming down. Once 10 is getting activated, thrombin is getting produced, it's kicking in and it's telling other factors to activate other factors more, so that more 10 can be activated, so more thrombin can be formed. So thrombin is encouraging the more clotting to happen, but at the same time thrombin is also preparing that we need to slow down also later. So even if you cannot remember that whole pathway, I think this is probably easy to remember, that prothrombin, factor 10 gets activated, that leads prothrombin to form more thrombin. Thrombin helps in converting fibrinogen to fibrin. Fibrin forms a clot or cross-link by activated factor 13. Okay, now who can repeat this whole thing? How, what's happening? What factors, what all this whole coagulation thing is happening?
So like from start of both.
No, just this last slide that we talked about. Huh? Coagulation cascade, little bit.
So sir, we have prothrombin.
No, wait. Let her say. Okay.
So will I get to see the picture of the?
So the picture has all the factors named there. Then you can just say factor 10 gets.
I think first step is the factor 7 is activating the
Tissue factor,
yes.
right? Yeah.
Factor 7 activated, and then I think fibrinogen is activating or converting. No, thrombogenesis is activating to the. I got it.
But see, this is good. You know why I want you guys to repeat it? So you can appreciate that when professors teach, it's a difficult job, right? At least, right? Okay. Somebody else. Can somebody else tell me?
Factor 10 get activated, it converts prothrombin to thrombin, which then gets converted into something else.
Which then gets converted, which then activates the
Fibrinogen gets converted to fibrin, right? And fibrin strands, right? And then factor 13, activated factor 13 converts fibrin strands and make them cross-linked, and now the clot that was loose is getting properly cross-linked, right? Now so
What's the role of calcium in one of
so you need calcium. Calcium helps in this clotting cascade, like factor 4 that we didn't talk here. It helps in clotting these activated factors getting activated. Now, one thing is also, do you guys know hemophilia?
Yes.
What is hemophilia?
So it's a sex-linked recessive disease in which the blood doesn't flow and it's hereditary.
Yes. So hemophilia is blood loving, right? So you don't get clotting very easily. So for hemophilia, if you guys know, I'll be super impressed, but if you don't know, I don't expect you to know. What factors do you think are causing hemophilia? Like, if missing factors, which missing factors can cause hemophilia? That's very cool. Okay, 8 is one. So there can be hemophilia A, B, or C. If 8 is missing, then it's hemophilia A. The other one is 9 and 11. Okay, so if somebody does not have 12, or low of 12, clotting can still happen, I think. Okay, but 11 is important, and that's why it's one type of hemophilia. 8 is important and 9 is important. You know, those are for hemophilia. Okay, so this is just to give you an idea that what happens. What is factor 2? Prothrombin, right? So this is just showing that how extrinsic pathway, intrinsic pathway help in more activated factor 10. More activated factor 10 starts converting prothrombin to thrombin, and this is what this cascading effect happening here, leading to clot formation. Negative feedback. So prothrombin helps create plasmin from plasminogen. Thrombin also stimulates antithrombin, to which heparin can bind. Heparin can bind to antithrombin 3, which can decrease prothrombin production. Thrombin binds to thrombomodulin on endothelial cells, which activates protein C, which also reduces thrombin production. So it's so cool. Thrombin is binding to thrombomodulin on endothelial cells, which is activating protein C, which is actually hurting thrombin production. Right? So these are the feedback loops that the clotting cascade has to slow down clotting slowly, so that it's just not keep going on and on and on.
It's a self-reinforcing feedback.
Yes, yes.
It itself is
Yeah,
inhibiting itself
yeah,
to produce.
yes.
It's feedback inhibition or feedback repression?
It's trying to inhibit the future production of thrombin, or slowing down the production of thrombin.
Yes, it produces side by side like
Yeah.
it is activating.
Yeah, and see, antithrombin is getting produced. It can bind to thrombin, but in presence of heparin, it speeds up very fast. Now, this is another interesting thing, that during coagulation, plasminogen protein is trapped within the clot, and then slowly plasminogen gets converted to plasmin, and this plasmin can digest the clot and fibrin. So when clot is forming, the blood is already putting something inside that clot that later can slowly eat up that clot and clear the skin or the blood vessel. Because see, if there is a cut, blood is clotting that small area, I don't want that clot to just keep sitting there in my bloodstream. So slowly that clot is going away, it's getting digested, so that it's like now nice and clean. We cannot, oh, what happened now?
One thing is there, like if the cut is deep, so we want the clot to be there to heal that.
Yes, yes. So it's not that, I mean, it's taking time to dissolve. Of course, you don't want a clot to happen and then within 10 minutes it to dissolve. That's not right, right? Then bleeding will keep happening. Okay. Oh. Okay. Okay. Thank you. So we cannot play video because of the online thing, but I highly recommend you watch this video. It's explaining whatever I said, but in a nice cartoon form, so it can be a nice video to watch. And you are getting these slides, I think. I hope Neha makes. Yeah, so she will send you more. Yeah, so. Okay. So again, hunger. Oh, okay, good.
And she will after the class? And she will after the class?
Okay. So calcium is required for reaction in coagulation cascade and for platelet activation. Thrombin is key element in common pathway. We talked about it. Prothrombin getting converted to thrombin. Platelets rapidly adhere to foreign surface, play role in intrinsic and extrinsic pathway. Activation happens within seconds for platelets. Clotting factors, factor 10, and deficiency in factor 8, 9, and 11 can cause hemophilia. Okay.
So only in the last line, in factor that is 12 or, so can you just reverse it? Yeah, in except 12 and 11, we need calcium to activate every other factor.
Other factors, yes. Okay, so another thing when we work with surfaces, because a little bit we are going to start going into biomaterials and medical devices and clot formation. Another thing to keep in mind is hemolysis, which is basically rupturing of red blood cells. So you can imagine that when we are testing our biomaterial, we don't even want to just test, oh, my material is preventing or reducing platelet activation or clot formation. We also want to make sure that our blood in contact with that surface is not causing cell lysis or red blood cell lysis, right? Another thing, so when we work with blood, you have to work with blood very, very gently. You know, you cannot just take a blood vacutainer or something and shake it, because by doing all this, you are really activating the platelets. Even when I'm drawing blood and transferring it to another container, you are not just dropping the blood, you gently put the blood so it slides down rather than just drop, because dropping this blood you might be activating the platelets. So you have to work with blood very, very gently. Hemolysis, non-thrombogenic damage resulting in cell death and release of components which can actually lead to more thrombus formation. Sometimes we have seen, like, if blood was not drawn properly or somebody had really mishandled blood or shaking, when we do this centrifuging of blood, you see that first, like your plasma should look very yellow, like the second picture, normal. But if it starts looking very reddish tint to it, that means the red blood cell lysis has happened, and that's not good when we are working with blood. Another thing, since we are going to talk a little bit about materials now, when you draw blood, for all these clotting, platelet surface interaction type studies, we want to use the blood within first 2 to 4 hours. The longer the blood is sitting, the platelets are a little bit dying or their activation capacity is going down, proteins are getting denatured. So you cannot, you should not do 10-hour study with the in vitro blood that was just sitting. At least for blood material interaction type studies, because platelets, proteins, fibrinogen getting denatured or not that effective, platelets are not that effective, activation might not happen so nicely, so you have to be careful with how old your blood is. When our lab does any of these blood material interaction in vitro studies, we get fresh blood and then within half an hour we start working on it, because sometimes we work with whole blood, sometimes we work just with PRP, because when we are testing blood material interactions or clotting, platelets and plasma are bigger problems or big players in the field, not the red blood cells. So we can separate red blood cells and we just work with PRP. So we centrifuge the blood, draw PRP and start working with it. Okay.
So what is PRP?
Platelet rich plasma. Okay.
And so can we do the gas chromatography for blood? Like we take the blood samples
So,
and try.
so we have blood gas machines to get what is oxygen or carbon dioxide to measure those things. Yes. So when we do animal studies, there is a blood gas machine where we can inject the blood. You need very little amounts to get all those parameters for blood. What is the pH of the blood, animal to animal, or what if your coating is causing pH change or something. So we want to do all that.
So we inject the blood like right away from extracting it?
Yes, so we want, when we do those type of studies, we do it very fresh blood. We don't let it just sit and do it later.
Okay, so what if we take the blood samples on a paper strip or something and then do it? Will it change the nature of results that I'm getting?
I'm not sure, probably yes, because what we do very fresh blood. Maybe that's a good study for you, right? If you are doing anything like that. See, whenever you have a doubt like that, so let's say my blood gas machine, I draw blood, why don't I inject little bit immediately, get the numbers, keep the blood for 1 hour, 2 hour, 5 hours, and then inject it again and see how much your numbers are varying. That can be a good test because I don't want to just say something because I don't know, because we always do it fresh. So what was I going to say? I forgot. Oh yes, so I was saying we need PPP also, or platelet poor plasma. Who can tell me, like, so let's say I'm centrifuging the blood, separating PRP with all these blood components. I have PRP. Why can't I just work with PRP? Why would I need to get some PPP also? It's a difficult question, but I want you to think a little bit, and it's okay if you cannot answer. So let's say I draw blood from a pig today or a rabbit, and I centrifuged it. I got my PRP. I can just take that PRP and start doing my studies. Okay. Why do I need PPP also? As a control, you are almost there. Can you explain what you mean as a control? See, my control can be, let's say I made one surface that is unmodified control surface, and then my surface that I have put heparin on it. Now I can put that PRP on this surface, PRP on this surface. After 1 hour, look at how many platelets are on this surface, how many platelets are on this surface. Why do I need PPP?
PPP contains less, like 10 times less amount of platelets.
Yes.
So that's why when we use PPP, exactly, I guess it gives us more chance because what do we say? The blood clot formation chances are less, I guess, and the heparin works much.
It would be acting as a negative control.
Okay. Something else? Okay. Anybody else?
Can be that with PPP we can study certain group of patients or certain samples where we want to look on the perspective where it's delayed coagulation or something.
Okay, no.
So can you have the question once again?
Okay, so maybe I'm not explaining it well also. So let's say I want to test a medical device that how much it's preventing platelet activation. Okay, so I got my fresh blood today. Can I just centrifuge it, get PRP, this yellow solution, and I have two surfaces that I'm comparing. I put some PRP on one surface, put some PRP on another surface, wait for let's say 1 hour, and then look at which surface has more platelets, which surface has less platelets, right?
Yeah.
That's with PRP. But I was saying we need PPP also. Why do you think we need PPP? What can be the use of PPP?
So it can reinforce the first results that we are getting. So amongst the two surfaces, let's say the one in which the PRP was less, it was less platelet rich, and since it was less, so the tendency for it to attach on the device would be lesser, as the concentration is lesser. So in contrast to this, if we take a platelet poor plasma, we can reinforce the same result that even at the more lesser quantity of it, we are having the same result. So it's like double cross validation or something.
Okay, okay, let me tell you. It was a difficult question, so it's okay.
Sir, maybe because in body plasma, platelet content is not constant. That's why we are trying to get that range of result.
You're almost there. Okay, so the reason is, let's say today I'm working with one animal, right? Now that animal has 4 10 to the power 8 platelets per ml, right? So I do my study with that platelet count. But you need to do always in science, you should always do replicate. N of 3, N of 5, N of 7, whatever, right? Tomorrow when I got the blood from another pig, that pig's platelet count is let's say 3 10 to the power 8, not 4 10 to the power 8. But your study on day 1 was done with one platelet count, on day 2 is done with another platelet count. So what we do is we get PPP so that we can always dilute our PRP to some concentration. So let's say the range of platelet counts in rabbits is 2 to 4 10 to the power 8. So I will always dilute my platelet count to 2 10 to the power 8. So today's rabbit was 3 10 to the power 8. Using PPP, I'll dilute it to make the concentration 2 10 to the power 8. Tomorrow's rabbit is 4 10 to the power 8, but with PPP from tomorrow's rabbit, I'll dilute the platelet count to 2 10 to the power 8. So now every day I'm working with the same concentration of platelets. It's not varying day to day from animal to animal.
We can call it true major baseline.
Yeah. So that's the reason. And the reason I asked you, I knew probably you won't be able to answer it, but the reason I asked you was, since you had to think about it, now you're not going to forget it. Right? So, and this is just example from blood. When you're designing any experiments in your research, always keep these things in mind. See, when I work with junior PhD students, sometimes they don't think about these things, and not their fault. I was like that also. Later on, when it's time to publish the paper, then you start realizing, oh, I didn't do this. Oh, my one study was, when you're writing and you're thinking more deeply, then you start realizing this mistake. Oh my, this study was done with 3 10 to the power 8. This study was done with 4 10 to the power 8. How can I draw a graph now? So just think very deeply before you are planning your experiment. Okay. How can we design hemocompatible materials? Okay. Hemocompatibility is important, right? Because if I'm designing a catheter or a stent or a vascular graft or a tubing for ECMO, I don't want blood to clot on my foreign surfaces. So I want to design better surfaces that won't clot. Normal healthy endothelium does not induce coagulation, right? It does not clot. It is perfect non-thrombogenic blood contacting material. Now, the beauty of nature of our body is tiny blood vessels don't clot. And we are working for years and years, we means not just me, so many great scientists, and we cannot make a non-thrombogenic surface. It clots sooner or later, it starts clotting. Key questions about designing blood contacting materials. What type of events tend to occur at blood material interaction? So see, application is important, translation is important, but mechanistic studies are equally important. If somebody won't understand how those interactions happen, how can we design materials without that knowledge that somebody did those studies? Because somebody was saying a day or two ago, they don't like mechanistic studies, they want something coming to market, which is what I like too. But if somebody won't do those mechanistic studies, it won't help us to bring something to market. How could these events affect patient or device? What are general ideas of the blood compatible biomaterials? What are major areas of research going? We will talk a little bit today, little bit we'll keep talking in the next few classes. Now, blood surface interactions. Some devices are only for minutes to hours, but some devices are for lifetime. And there is different material design involved with different types of applications, right? Heparin is used clinically, that's a standard right now, but even with heparin, sometimes very difficult to prevent clot formation, right? All that black you are seeing, that's all clots in those oxygenators or tubings or catheters, that's all clot formation. Also, heparin can be immobilized on the surface, heparin can be injected in the patient, but I think some percent, very small, 5% or less patients are allergic to heparin also, and it can cause heparin induced thrombocytopenia or HIT. That's why we want to design new materials, new surfaces that can prevent all this from happening. Blood flow, conditions of blood, length of time, mechanical properties, all that needs to be considered when you're designing a surface. Active materials or passive materials, active or passive coatings, that's we need to think about also. What is getting absorbed on the surface, effects of adhered cells or cellular components, how immune system is going to do when it happens. Is your material going to be toxic? So if you put a coating on your catheter, it's going to go in your blood vessel. Is that going to leach something, or when it's touching your tissue, is it going to kill any healthy cells? So we need to think about cytotoxicity, we need to think about hemolysis, and while doing all that, its main purpose is it should prevent clotting also. So that's why you can imagine so difficult to make a device to come to market, because all this has to happen at very low cost also. So many challenges. Material should be stable at room temperature also. It can be sterilized also. It can be stored also. It is not toxic. It's not hemolytic. And it's not expensive also. So now in simple language, we were talking more complicated. In simple, fibrinogen gets absorbed on the surface. Let's say this is a catheter surface. Platelets come and adhere to it, get activated. Once the clot starts forming, fibrinogen gets converted to fibrin. Fibrin is getting cross-linked. Some red blood cells, some white blood cells also get trapped in that clot, and this clotting is happening. And now you also know, because of coagulation cascade, Once this coagulation starts happening, more and more platelets are coming. See, it's not just little bit happening. When these platelets are getting activated, it's telling their neighboring platelets, come on, we need to clot on this surface. And now this clotting is just going out of control. Blood is very complex mixture of so many different proteins, red blood cells, platelets, lipids, carbohydrates. We also talked about proteins rush to the surface, albumin comes first, fibrinogen comes, replaces albumin, that's called Roman effect. All that is happening. Here is little bit showing different amounts of different proteins and their diffusion coefficient, what is rushing fast, what's coming slow, how different proteins are coming. We already said albumin comes, later on fibrinogen comes, some proteins interact more with the surface, some proteins interact less with the surface. Material itself is non-thrombogenic, but then when surface concentrations of proteins change, platelets get activated, it can start causing more platelets to come, clotting to start happen. Okay, now when we have, this is just showing a little bit, don't worry about it, but one thing is, this is again Dr. Buddy Ratner, Dr. Hoffman that we talked, it's their third edition book, I think, from where we got this. So how intrinsic and extrinsic pathway, factor 10, thrombin, prothrombin, it's all that. But in our body, in our endothelial cells, there is something that's preventing from this clotting to happen. And some of those things are protein C, heparin-like material, heparin sulfate, nitric oxide. So those are good things. Prostacyclin, those are good things present in our endothelium that's not letting clotting happen all the time. So one easy thing is to start learning from there that, okay, these are the things that help and prevent clotting from happen. Okay. So prostacyclin, it prevents platelet activation, vasodilator. Nitric oxide, we will talk a lot about it. Heparin, we know heparin sulfate and heparin, it prevents clotting from happen. Protein C, we also said when thrombin starts binding to the endothelium cells, it increases protein C, which starts inactivating clot. Now, in a little different way, we are again showing what we little bit talked about earlier, how it's basically same, that extrinsic and intrinsic pathway, how platelets are contributing on injury side, plasma contributing initially for artificial surface, but then everything gets mixed together. Heparin works with antithrombin 3, which binds to thrombin, which prevents clotting. What's missing here is nitric oxide, which acts here. Because nitric oxide is preventing platelet activation, right? And we are going to talk about that in future work. So one thing that we are doing is, why not design surfaces that are not just preventing platelet activation, that are not just preventing protein adsorption, but are preventing platelet activation and protein adsorption both, right? Together, synergistically.
So what do you need to prevent both? Platelet activation and
Protein adsorption. To make anti-fouling surface, right? Because if we prevent platelet activation, that's great. But protein is still getting adsorbed. And that's making very difficult for our coating to keep fighting platelet activation because there is a layer of protein that's attracting platelets. Our nitric oxide or whatever component is fighting platelet activation, but there is a component protein sitting there that's calling platelets, so it's more difficult. But if you do both, now the chances are getting lesser and lesser of clot to happen, right? Okay, this got a little bit. Okay, now there are a lot of different types of coatings. So coatings can be passive coatings or active coatings. Okay, passive, like the name suggests, it's like trying to prevent things to stick to the surface. So for example, when we are making non-fouling surfaces, we are trying to make hydrophilic surfaces or very hydrophobic surfaces, so things don't stick to it. Polyethylene glycol is a simple example. Zwitterions, they are coming later also, but that's like non-fouling. You're preventing things to stick. You can attach endothelial cells because see, when endothelial cell layer is there, blood is just flowing nicely on endothelial cells. When somebody get a vascular graft or stent, you want endothelial cells to grow on the surface because it's almost a permanent implant. So now if stent is there, the sooner it gets covered with this endothelial cell lining, better it is, because now blood will keep flowing on it without getting activated, right? So you can immediately think, oh, so on my device I can grow endothelial cells and then implant that. But challenges with cell seeding, proliferation, stability, because imagine, let's say stent, and I'm not a surgeon, but you can imagine, you know how stents are deployed in the body? Okay, so we will talk about stents later. We cannot watch a video, but what they do is, so let's say there is a guide wire. At the end of the guide wire, there is this balloon, and all this I'm talking is like very tiny, very thin wire that's going to grow in your blood vessel. Okay, at the end of the guide wire, there is this balloon. You guys know how stents look.
So that mesh figure you
The mesh type of thing, like, you know, this is like a metal mesh or something, you can think. Yes. And in future, next week, I'm going to show you stents a little bit more. So let's say you put this stent on top of that balloon, and now this wire is going in the blood vessel at the location where the clot is there, and then you are inflating that balloon from outside with the syringe, so that with the air, balloon inflates. When it inflates, this metallic stent, it opens up or it gets deployed, and its structure is such that once it opens up, it won't close, which is what we want, right? So now you can imagine it's so complicated, right? You have a balloon, you're putting a stent on it, you are taking it to that location, you are inflating the stent, then you're deflating your balloon and you're bringing the wire back, and stent is now fixed in the blood vessel. If you have all this cell seeding going on it, it's so complicated, right? And then before you implant any medical device in the body, it needs to be sterilized also. So can we sterilize all these endothelial cells? Also, stents, hospitals can buy and keep it. This type of cell seeding type thing, you need to order it the day of surgery, it needs to arrive, and then you need to put it. And it just sounds, it's going to be very expensive. A stent that you're making versus this type of seeding on the, it sounds it's going to be very much more expensive and complicated to work with, right? So this is a challenge with that type of application. Yes.
So why is stents inserted?
Oh, so if somebody has a clot, so let's say somebody has a clot or the blood vessel is narrowing
Anywhere in the body.
in the blood vessel, yes. So then you have to put us, because if the blood vessels keep narrowing,
Constrict the blood.
now it can be a heart problem or stopping of blood flow. So you put a stent to open that so the blood can keep flowing, right? Now that I just took that example, but the same example is for catheters or for vascular grafts. Now other things we can do is mimic endothelium layer. You want ultra smooth surface, you can have slippery surface, negatively charged surface, zwitterionic surface. Zwitterion, the net charge is neutral, it's not positive or negative. Or you can have active release of nitric oxide, which we'll talk a lot later. Or you can have immobilized heparin on the surface, which products are already in market. Each one of these have challenges associated with it that we will little bit talk more about. But these are some of the big things researchers are trying. Maybe the new zwitterion is coming, new nitric oxide donor is coming, new way to make a slippery surface or super hydrophobic surface. All that is little bit changing, but some areas that are going on right now. Okay. Then
the material by using which we make stents, why don't we make a catheter of that material? Because you said that stents usually don't it allows the blood flow and the blood don't coagulate on the stent. The validity is more than the catheter.
The blood won't. No. I never said blood won't. See, the goal of stent is stent has a equal. Everything has a challenge. If you put anything in blood, the clotting can happen. We do not have a truly hemocompatible material, right? And stents are a lot of time metal. Catheter, we want a flexible tubing, so it's polymeric. But stent, no way it's a hemocompatible. I mean, we are trying to make it hemocompatible, right? In stents, we want endothelial cells to grow on them so that it gets covered. Catheter, we don't want that because catheter will be removed after few days or weeks. It's temporary, right? No, I think it's fine. I was just, yes.
So, is there any current research going about carbon nanotubes or something?
Everything is going on. I'll show you elastin. You talked about elastin. After this class is over, I'll show you. I found a paper on elastin also. So whatever you can think about, somebody is working on it. Okay. Okay. A little bit more. Okay. So when we work with blood, it's very complex and very fragile, like we talked, right? We have to just treat it very gently. Blood handling protocol. So in our lab, we have a proper SOP. The day when you draw blood, how much heparin is there added or any anticoagulant is added in the tube, who will go draw blood, how will we bring the blood. People who are working with blood, they are properly trained. How much time we can store it, like only few hours, 2 to 4 hours. What blood flow rate we will use, we will talk tomorrow. You can work in a 96 well plate or a small well plate, or you can have an external circuit flowing blood and your device is inside that circuit, more mimicking human body. Donors, we are not working with human blood, But if somebody is working with human blood, then age of the patient and NIH is very careful now about if you are using male or female, you need to really justify, and if there is no proper justification, you should have equal number of male or female donors. Design of device, we little bit talked about how surfaces change, coatings, roughness, all that will play a role here. If you are not making a good surface, nice coating, even if you have a good component or release of heparin or nitric oxide or zwitterion, if your coating is not properly done, it's going to activate blood. What anticoagulants to add? So see, we also have to keep in mind that in vitro studies are great, but the challenge, one challenge with, what is one challenge with in vitro studies with blood?
So it's not in the physiological parameters.
It's not in physiological parameters. What else?
So the homeostasis is disturbed.
Yes, so after we draw the blood, that platelets, proteins, they are dying, they are not like as like a fresh blood.
Vessel pressure is not maintained.
Yes.
The blood pressure is not maintained.
Yes.
The blood flow.
Blood can coagulate.
Blood will 100% coagulate, but to prevent blood to coagulate, immediately. So if I draw your blood, you were not here, that was the first thing we were talking. If I draw your blood right now in a syringe without adding heparin or anticoagulant, citrate or whatever, blood will coagulate.
This can be the problem.
Okay, so to prevent this, what should we do?
So we can keep it running.
No, but if I want to do in vitro study, I'm drawing blood. Now blood is going to coagulate. So what should I do?
Anticoagulant.
I add anticoagulant. But now if my blood has anticoagulant and I'm testing my control material and my material,
We would never get the real result.
I have added anticoagulant to it. So sometimes we reverse the effect of anticoagulant also, right? But now the problem is, if you reverse the effect of anticoagulant, on control, if the clotting is happening really fast, but here or your main surface, you are going for an hour. Now after an hour, when you look at the surface, how many platelets got activated or what is clot, it will look very difficult for us to prove it, because on one we see this big clot, but in real life if you put a catheter in an animal, it's not going to be such drastic that full clot and no clot. You can at least get a 4-week data and compare the surfaces better. Now if I draw blood with anticoagulant, the coagulation is not going to be proper. If I reverse anticoagulant on my control surface, very bad coagulation can happen very rapidly. So how to play with that? Yes.
So can we just not replicate the body parameters and keep blood flowing via vessels or tubes like that with the same temperature, same pH and the same dimension of the blood vessel so that the blood pressure is equally maintained and then test the device?
Wow, that's such a dream, right? That's isn't that why we are designing these devices, that nothing clots on the tubing? See,
If you are using ECMO machine,
yes, so there we have to add heparin to the blood, right? So you have so much heparin, that's preventing anything from clotting, right? But if we want to go away from heparin or reduce heparin, we need to design surfaces that will prevent any clotting to happen.
That's like paradoxical.
Yeah, yes.
like our blood vessels are like the blood vessels is made up of some the biomaterial. If we can just make it in an artificial manner,
That's what we are all trying, right? Like the slide I showed you, I don't want to move, then it'll hang up again. So endothelium layer, it has all that prostacyclin, nitric oxide, heparan sulfate. That's what we are learning, biomimicry, right? We are learning what's there, what's preventing. We are making surfaces as close to that as possible. It's working, but it's still very challenging. You still have to make it work in those constraints of cost, sterilizability, storage stability. If you put a really amazing coating, but the coating cannot be survive more than a day. I mean, seriously today, go home, search on Google Scholar, hemocompatible materials, hemocompatible surfaces. You will see there are so many publications, so much good work is done on this, but we still need to give heparin to patients. Still the tubings clot, still the catheters clot.
So one mechanical tweak, like on the, irrespective of the material of the device, obviously we'll take hemocompatible metal or something. But sir, as you said, like one platelet has like 60 to 80 thousands of those receptors. So in order for that to settle on the device surface might be having pores, compatible to those receptors fitting upon it. So what if we create such a smooth device which has pores very small, way comparable, like 10 to the power 2 or 3 times smaller than that? So would it still stick?
Protein sticks.
Okay, now
Fibrinogen.
blood won't, but protein would.
Fibrinogen is the first thing that sticks.
Okay.
Right? I showed you a cartoon that as soon as you put a device, within seconds to minutes, protein will get stuck. And then, see, yesterday our whole talk was protein, but platelets is so exciting that you forgot protein, that's another culprit. Platelets are one culprit, but protein is also a culprit. So you are fighting against platelets and proteins.
So like we have 150 some proteins roughly. So we must have cataloged the data that what all receptors probably
So
are
the
like.
big problem is fibrinogen.
Okay, it's the only major culprit.
One of the big, for platelets to get clotted on the surface, yes, fibrinogen is the biggest culprit.
So we have to prevent anti-fouling, it should be, yeah.
So it's a very small problem,
But it's a very
kind of easy to explain problem, but the solution is very, very complicated. But it's an interesting problem, right? Okay, so we can stop here, but I can still answer your questions. We can keep the discussion going.
So like we have this pacemakers. So how long does it work? Apart from that battery gets over in a decade or so and we need to reimplant it. Like what material is it made of?
So I think pacemakers, people have it maybe, I think for a very, very long, like for after years they just change the, yeah. So yes.
So what is the material it is made of?
So I am not 100% sure, but I think these leads have some covering on it, and infection I think is a bigger problem.
Oh, it does have.
For pacemakers, infection is a big problem, I think. I think so. I might be wrong, but I think infection is a bigger, because if you have any tubing coming out from your body, there is an interface of that can be
So but
caused.
pacemaker, I guess
You put it under yes.
after the surgery it's
Yeah.
completely.
So I don't know how much clotting is a problem with pacemaker.
Okay,
Yes.
so why not, but it's constantly, although it's giving the electrical impulse, but is it not washed in blood all the time?
I think it's sitting outside the pocket, right? It's not.
No blood
It's.
is draining the pacemaker then.
I think so. I don't know if blood touches the pacemaker. I feel it's just sitting not inside the blood vessel, it's sitting outside, under the skin.
So we would have vessels and blood rupturing it.
Maybe during that surgery or when you're putting it, you're giving heparin to the. So whenever these vascular grafts, stents, these type of surgeries are done, patients are given heparin for sometimes for years, because if somebody has a stent, they are given heparin for a long time. But if you want to go away, but then clotting can still happen. So we want to just go away a little bit from heparin or reduce heparin so that we don't have to worry about giving heavy doses of heparin. Yeah, sorry, yeah. Oh.
You are saying that we give an amount of heparin for a really long time. So, is there any problem with these high amount of heparin?
Exactly my question.
With heparin?
Yes, sir, because
Yeah.
I mean inside body it's one thing, but we're giving heparin from outside of the body, so
Yes.
it's external.
And
So
other thing is that like we are adding it into the blood, so in some way or the other, the composition of the blood is also somewhat changing because we are adding heparin, a different substance.
Yes. So heparin is standard that's given during surgeries and all these treatments, ECMO and all that. The main problems with heparin, one, even after giving so much heparin, clotting still happens. I showed you one picture where this oxygenator with black. So clotting is still happening. Second, if somebody is allergic to heparin, it can cause thrombocytopenia or hit heparin-induced thrombocytopenia. Third, if somebody has heavy amount of heparin, if they get injured, their blood is so thin now that the person can actually bleed. So that's why it's needed, but if we can, even if our coating is not 100% preventing, but it's still reducing a lot clotting, maybe we can reduce the dose of heparin. We don't have to give so much heparin to the patient now.
So, but then it will clot inside that system. Like if we are using ECMO.
So if our system is all coated with whatever works, we will show nitric oxide, but whatever coating works, now you don't have to give, because I don't want to tell you everything about nitric oxide today, that's next week, but that coating is just working to prevent clotting and happening on that surface. It's not impacting your blood. So if you get injured and if you have a catheter that's nitric oxide coated, you're not going to bleed. That nitric oxide or that coating is just acting at that interface. It's not impacting your blood in the body, so now your blood is thin and you can actually bleed.
just a suggestion, because I just saw a post that scientists made similar heart cell wall like stems from stem cell in a petri dish, like outside of the body.
Yeah.
So those muscles are like doing
Yeah.
this. So if we can make blood vessels outside the body or in a petri dish and use those vessels as pipes for different machines or anything, so can it will
I mean, it's a
we
great
need another
idea, but
coating
you
for
can
it.
But you can imagine the complexity, right? Where we wrote endothelial, like seeding of cells, sterilizability of that material, storage stability of that material. Can it be shipped so easily?
Because first it need to be grown and then we need
and what will be the cost of that, right? Even when I talk with companies for urinary catheter or whatever, if a catheter is sold for $10,
same thing.
even if your catheter is much better but it's 5 times the cost, it will be very difficult to bring it to market. Okay guys, see you tomorrow. We will maybe finish this blood talk and start talking about bacteria and infection. So fouling was one problem, clotting is one problem, infection is next problem, and then from next week we will start talking about some solutions. And I want to give you a little bit tips about PhD. I want to talk a little bit about ethical concerns in science, which is big topic. You know, we have to do good research, but we have to follow the ethics. You cannot try to find shortcuts. Ethics should be number one. So we will talk about that and little bit about academia versus industry, That can be interesting topic for, so please try to come next week also. I know some of you are coming first time today, so try if you are available to come next week also. Okay guys, thank you so much. Thank you. Thank you. Thank you. Thank you. Okay, you have. Oh, so sorry, I forgot it's in my pocket.
So, thank you so much, sir, for that presentation. We are concluding day 4 after this lecture, and we have no lectures afterwards. And now we will be free for lunch. So, I request everybody to please proceed for lunch. Thank you.
Oh, you don't have a speaker today?
No, sir, we don't have.